All right, well, welcome everyone to this week's edition of the Extracellular Vesicle Club. You can see that I'm very excited about the session today because I actually wore a tie. So we're looking forward to a great presentation from Annalisa Radeghieri and Angelo Musicò. Annalisa is in Italy, which is where I'm going to be going next week for the ICEV EVs in the nervous system meeting. And Angelo is in Spain right now. But we're going to be turning our attention to something much more inward and on the nanoscale, which is the EV corona. And so research has progressed now to the point that we have multiple groups around the world that are looking at this phenomenon of these things that sort of loosely associate with extracellular vesicles. This corona research, though, it kind of goes back to the days of the synthetic nanoparticle research, where people were finding that there was a so-called hard corona and a soft corona that it can associate with nanoparticles, synthetic nanoparticles that are introduced into a complex biological environment like the blood. So we're going to hear a lot more about this, but I'm going to stop talking here and hand it over first to Annalisa, who's going to give us an introduction, and then we'll go to Angelo. So thank you both for being here today. Everybody, please put your comments and questions in the chat box, and then we'll allow unmuting at the end of the session. Annalisa.
Okay. Share my screen. Okay. You see my everything?
Looks great.
Okay, so good afternoon, everybody. It's really a pleasure to be here and to present, and thanks Ken and the AV Club folks for inviting us to present our manuscript, which has been the result of a strict collaboration between, of course, myself and Giuseppe Pomarico, who cannot be here right now for academic issues, and Paolo Bergese, and of course, of talented students like Angelo and Rossella Zenatelli in our lab. So just to introduce the topic, as you just said, Ken, so in the last decade, the field of nanoparticles, nanomaterials has really been completely revolutionized, since it has been understood that artificial nanoparticles, once synthesized, so having their own size, their own surface chemistry, etc. When they get in contact with biological fluid, they actually change their identity. So they change from their synthetic identity, from their structure, original structure, to a new actually structure and size and shape and aggregation state. And this in turn cause a novel kind of a different way of in that the particle actually distribute in the biological fluid. So in the way that nanoparticles interact with cell membrane, with other biomolecules. And this is has been mainly ascribed to the formation of what this so-called biomolecular corona. So this concept has been first coined by Sederval in 2007, but has been then developed in the lab of Dawson by also Marco Monopoli lab, where they could study a little bit more about these nanoparticles, and they found that indeed, as soon as a nanoparticle is immersed in a biofluids in a very, very short time, it gets surrounded by a plethora of biomolecules, which actually are absorbed on the surface of the nanoparticle itself. And in this way, these nanoparticles, they are actually continuously, these biomolecules are continuously exchanged with the biofluid, and they determine the formation of some layers. Some have described the formation of multi-layers on those nanoparticles that change completely the structure and the capability of the nanoparticle to interact with membrane and with other biomolecules. So we can talk about the formation of a hard or a soft corona, as you just mentioned. That means that either tight bondings, tight interactions, or soft, weak interaction can happen at the level of the protein corona, and this completely changed the characteristic of the nanoparticle. So this concept that has been first coined for synthetic nanoparticles, then in the latest years has been translated to the word of extracellular biogenic nanoparticles, that is for extracellular vesicles and lipoproteins. Indeed, many works, as you see here, many groups have already started to to tackle this point by simply evidencing that on the surface of nanoparticles, biological nanoparticles, there are some proteins that, or either biomolecules that are absorbed on their surface by weak interactions. So in the case of, for example, EVs, this nice review from Halal shows that EVs, either other than showing integral membrane proteins, they also show some they call interacting partner, that is many biomolecules, for example, proteins, but also nucleic acid that you see here, and also even other nanoparticles, lipoproteins, that can have an interaction with EVs themselves. And also, it's since I talked about different biogenic nanoparticles, also lipoproteins are covered by a absorbed protein, by a protein corona. This is very interesting to me because it has never been considered before, the fact that HDLs or LDLs circulating, they have a protein corona. So, and what is very interesting is that recently they have discovered, for example, that there exist more than 16 different types of HDLs, each bearing a different function, for example, antioxidative or anti-inflammatory function, or either some function in hemostasis, or of course in lipid metabolism. All these functions are given by the absorbed proteins that could be found on the surface of these nanoparticles. So this is very interesting. So for us, it is also very important to notice that what are the determinants of the formation of the corona on these nanoparticles. Well, first of all, we have to keep in mind that the interface of between the surface of the nanoparticle and the environment determines what will be the composition and the structure of the corona. So all the interaction, the bindings that happen at the interface of the nanoparticle will determine what will happen and what we're gonna find basically in the corona. And the second important point that I would like to state is that it has been shown that there is a selective enrichment of proteins and metabolites in the corona. So basically, of course, you can find in the corona only the biomolecules that you find in the biofluid, but not all the biomolecules that you find in the biofluid could be then found attached in the corona due to the specific enrichment. And so, and another important point is that sometimes you can find in the corona even low abundant molecules that cannot be seen in if you look in the total complexity of the biofluid. So what are the factors that influence this absorption onto NPs? First of all, as I said, the biofluid composition. So of course, different biofluids will change the protein corona on the nanoparticle. And second, the biomolecular and the biophysical properties of the nanoparticle themselves. So this opens really a big Pandora's box thinking about vesicles, because if you think about the huge heterogeneity of vesicles, small ones, big ones, microvesicle, exosomes, they all have a different membrane composition. So we may think that there's a corona, a unique corona for each vesicle that we have in our body because of their composition. And of course, another point is that protein properties, their post-translational modification or their mutation will change the way the proteins are absorbed on the surface of the vesicles. So this immediately comes give us the idea to think about the pathological condition. What about if the pH of the medium changes, or the protein concentration is altered in the case of a pathology? So this thought has led to the idea that really the corona can be thought as a new tool, a new platform for biomarker discovery, and to study also the pathogenesis mechanism of many pathologies that maybe have not been really understood. But not only this, today we're not going to talk about this. Actually, our group has been also involved in this topic in the diagnostic field, showing, for example, that plasma AV corona may help to discriminate healthy from antithrombin deficiency affected subject. Basically, we have shown that different glycoforms of antithrombin are absorbed on the surface of the vesicles, comparing healthy subject and affected subject. So corona can really be a tool to discriminate healthy from non-healthy subjects. But so this was the diagnostic point of view, but due to its feature, AV corona may impact also the application of AVs in therapy. Why can I say this? Well, you just have to think about, for example, AV surface engineering process or the AV administration in vitro or AV administration in vivo. So for these last 2 points, this has been made much more clearer for synthetic nanoparticles, because we all know that when a nanoparticle is injected in the body, that it immediately gets surrounded by biomolecules in the biofluid. So this dramatically changes the way the nanoparticles biodistributes, and also it's kinetic. So this would be also has to be translated also in the EV field when we want to use EVs for therapeutics. And the other point that we would like to point to make today is that so many groups are attempting to use vesicles to deliver drugs to, for example, to tumors. But since many vesicles have not the targeting properties that we look for, we try to functionalize them in different ways. So some ways to functionalize vesicle, they just use vesicle, they obtain vesicles and then they try to functionalize the surface of the vesicle with different ligands or with proteins. And if you think about it, then you simply have to imagine that the vesicle is immersed in a fluid in which all the proteins are present, the proteins that you would like to covalently link, okay, to your vesicle. But this is if you think about it, it's simply a biofluid. So all the proteins may, in principle, absorb to the surface of the nanoparticle and form a corona during this process. So in our lab, we started talking about this, and so the idea came to our mind that why don't we try to exploit the formation of AV corona for AV surface engineering? So why cannot we use AV corona simply to engineer our vesicles and perform targeting? And so to answer this question, we decided to explore 2 edge cases of EVs modified with an antibody, with cetuximab. And so we decided to analyze one case that is it's a chemisorption case, because in this case we simply covalently bound the antibody cetuximab to extracellular vesicles. And on the opposite case, we try to instead mimic the formation, or induce actually the formation of a corona by physisorption, by simply incubating the antibody with extracellular vesicles. And we use the antibody cetuximab because we were interested in targeting triple negative breast cancer cells, because they express very high levels of EGFR, so more than 90% of the cell express this kind of receptor. And so we started using cetuximab for this reason. And now Angelo will introduce what we have done in our lab.
Okay, well, hello everybody, and thank you for the introduction. Well, in this work, we use as a study model red blood cells derived EVs that we produce by red blood cells after chemical induction by culture ionophore, following literal protocols made by the group of Dr. Min Lee in Singapore. And after the purification of our EVs, we characterize them by determining the size distribution by NTA. And we found that we have a population, a monomodal size distribution in our EVs, with the peak at around 160 nanometers. We determined the morphology by AFM. We also have presence of a negligible amount of soluble protein contaminants in our preparation with the column assay, that is a colorimetric assay developed in our lab, that is based on the aggregation of gold nanoparticles onto lipid membranes. We also determined the ratio between particles and proteins in our EV preparations by respectively NTA and BCA. And finally, for the characterization of our vesicles, we assess the presence of EV protein markers such as LAMP1 and ALIX, red blood cells markers such as the hemoglobin, and very importantly, we also assess the absence of the EGFR in our EV preparation, as we aim to study the two edge cases of physisorption and chemisorption as engineering strategies for EV surface functionalization, and the presence of FGFR, that is the natural ligand of the cetuximab, the molecule that we want to bind to our vesicle surface, could also allow the possibility of an immunosorption mechanism. So it's very important that in our vesicle preparation, there is not EGFR. So for the chemical strategy, we use a two-step chemical labeling approach that is commonly used for the surface engineering of vesicles with big molecules, such as indeed an antibody. And this approach exploits the orthogonal chemistry, so a set of chemical reactions that can take place in biological environment without affecting biomolecules, as
Angelo,
it involves
sorry to interrupt.
not naturally present groups.
Sorry to interrupt.
Yeah.
I think the slides are not advanced.
Sorry, Ken?
Here we go.
Oh, this is good.
Okay. Okay. Yeah. I was saying that for the chemisorption strategy that we use in this work, we followed a two-step chemical labeling approach that is commonly used for the functionalization of EVs with big macromolecules such as antibodies. And in this approach, we use the orthogonal chemistry, so a set of reactions that take place in biological environment without affecting biomolecules. And this orthogonal chemistry reaction don't affect the biomolecules, as they involve not naturally present groups, such as the azido groups and alkyne. And the other reaction that we use to functionalize our building blocks, so the antibody, can you skip the slide, Anna, please? Sorry. The second type of reaction that we use in this work for functionalize the antibody and the AVs in order to react them by the orthogonal chemistry is the coupling between an amine compound and an activated ester. So we exploited the amine presence on the side chain of lysines in the proteins in the case of the antibody of the cetuximab, to synthesize the modified cetuximab. And so we react the antibody with a fluorophore to track it, and with the BCO, so the alkyne, to give to the antibody orthogonal reactivity toward the azido group that with the same chemical reaction we attach on the vesicle surface. Okay, next slide, please. Okay, so next we designed the simple protocol in order to explore the possibility of physisorption and chemisorption, so the two edge cases that we are considering. And in the first case, so for the chemisorption, we firstly react our vesicles with the azido PEG, and after the purification from the excess of PEG, by filtration, we react them with the modified antibody, and the BCO for the reaction with the azide. And then we purify our chemisorptive EVs by ultracentrifugation. While for the physisorption strategy, we directly react our EVs with the modified antibody, and then we ultracentrifuge them in order to see if there's also a physisorption. So then, after the purification, we characterize our EVs. And firstly, we determine their site distribution by NTA, and we assess that the functionalization process in both the cases didn't affect so much the site distribution of our vesicles. Indeed, we have a site distribution that is overlapping with the ones of the pristine EVs. And again, by the Conan assay, we assess the presence of a negligible amount of free antibody. And this is very important for us because this indicates us that all the antibody that we are going that we are going to quantify in the next steps is mostly related to the EV surface. So we denaturated our samples and we ran them in an electrophoretic gel. And following the fluorescence of the antibody, we quantify the amount of antibody in our vesicles by building a calibration curve with the standard MCTX. And then we normalize the molar concentration of the antibody that we have quantified for the molar concentration of EVs in our preparation. And we found that in both the cases, for both the cases, for the click EVs and the physisorbed ones, we have the same amount of cetuximab per EVs, that is around 85 molecules of antibody per EVs. But analyzing the densitometry profile of the electrophoretic run, please, Annalisa, next slide. So I was saying that analyzing the densitometry profile of the electrophoretic run of our functionalized vesicles following the fluorescence of the antibody, we find that in the chemisorb DVs, we have smeared signals around the lane that and a different band pattern compared with the standard CTX, the standard antibody, indicating actually equivalent binding of the antibody with other proteins that cause indeed a change in the molecular weight that it's is the reason why we see a different pattern. Indeed, and on the other hand, for the physisorb DVs, we see that the densitometry profile of the lane is identical to the one of the standard antibody, indicating that in this case, not covalent interaction occurs, and we have just the antibody. And so, go ahead, please, Ken. So then we try to optimize our protocols and to evaluate the stability of our functionalized EVs. In this case, I'm talking about chemisorb EVs. So we try to react our vesicles with increasing equivalence of MCTX, and we evaluated the stability by washing several times, three times, by ultracentrifusion, and after every wash quantifying the molar concentration of antibody per EVs. And in the case of the chemisorb EVs, we see that after every reaction ratio and after every washing steps, we see that the amount of antibody per EVs is constant, indicating that indicating two things. The first one is that already at the first reaction ratio, we have saturated the surface of our vesicles with antibody. And the second thing is that the binding is stable and covalent. Indeed, after every washing step, we didn't lose antibody. And interestingly, we also calculated the theoretical surface coverage of the antibody on EVs, approximating the antibody as a sphere of 7.5 nanometer of radius, and the EVs at sphere of 160 nanometers of diameter. And we found that after this calculation, we found that the surface coverage theoretically is around 10%. That is pretty low, but it seems that we have already saturated the surface, and also that we don't have physisorption in this case. Indeed, we don't lose antibody. While in the case of the physisorb samples, next slide, please. We see that increase in the reaction ratio. We see also an increase in the co-isolation yield of the antibody per EVs, but after every washing steps, we also see a loss of the antibody while it seems that the physisorbed antibody on the vesicles seems to be stable at a surface coverage of around 5%, thus indicating probably the formation of two types of corona made by the same molecule. Indeed, it seems that at surface coverage high surface coverage at around 30%, we have a soft corona, so weak interaction of the antibody with the vesicle surface, while at a surface coverage of around 5%, the interaction is much stronger, and it is retained after the ultracentrifugation. And next slide, please. And so, resuming the results of this part, we demonstrated that with or without click chemistry, the cetuximab associates on the vesicle surface, but with the click chemistry, a stable coverage forms, while performing physisorption resists to the formation of a classic protein corona, with also a suggestion of an hard and a soft corona. Next slide. So we start asking if does the chemisorbed cetuximab or the physisorbed ones can work in the same way at a molecular level, and after that also at a cellular uptake level. Next slide. For evaluate the affinity of our functionalized EVs toward the EGFR, we performed the surface plasma resonance. We mobilized on the strength-coated chip the EGFR, so the natural ligand of the cetuximab, and we tested EVs with the same functionalized EVs by chemisorption and physisorption with the same amount of molecules of cetuximab per EVs, that is around 85. And as you can see, the binding curve that we obtained testing these two samples are overlapping, and with an apparent affinity constant that is around 0.2 nanomolar, 1 order of magnitude lower than the affinity constant of the free antibody, indicating that probably there is a cooperation mechanism due to the multivalence of the bounding of the bounded antibody on the vesicle surface, and that lead to an increase of the affinity toward the EGFR. As we can see, on a molecular level, it seems that the two types of EVs work in the same way. So we move on the cellular uptake test. And next slide. So to evaluate the cellular uptake of our functionalized EVs, we test them against the triple-negative breast cancer cell model, the MDA-MB231 cell line. And we evaluated the uptake by quantifying the fluorescence of the membrite, the hydrophobic probe that we use to label our EVs. And we quantified this fluorescence by confocal microscopy. And we test our EVs after 4 and 24-hour of incubation on the cells, and in presence or in absence of FBS, so in presence or in absence of an high concentration of soluble proteins. And so, as you can see, after 4 hours of incubation, we see that there are no differences between the uptake of functionalized EVs and pristine EVs, but at 24 hours of incubation, we can see that in absence of FBS, functionalized EVs, both chemisorbed and physisorbed ones, have the same uptake that is higher than when compared with the pristine EVs. But very interestingly, in the presence of FBS, we can see that only the chemisorbed EVs have a higher uptake compared to the physisorbed ones and the pristine. And while for the physisorbed EVs, we see that they have the same uptake of the pristine EVs. And this data is very interesting because strongly suggests an exchange of macromolecules at the surface of our vesicles due to the presence of the FBS, so of a higher concentration of soluble proteins, which can lead again to this exchange of macromolecules at the surface, leading to a loss of functionality. And next slide, please. And also, indeed, we tested, we quantify also the fluorescence of the cetuximab in order to relate its uptake on the cells. And again, after these data are related to the 24 hours of incubation. And again, also in this case, in the absence of FBS, we have a very similar uptake of the cetuximab in chemisop, physisop disease. And again, in the presence of FBS, we have chemisop disease with a higher uptake than of the cetuximab compared to the physisop one. And next slide, please. And indeed, we, as I said before, this data strongly suggests that the presence of the FBS lead to the exchange of macromolecules on the vesicle surface by forming another protein corona. Indeed, as Annalisa told before, the corona is always related with the environment, the nanoparticle environment interface. And in this case, so we expect that macromolecules of the FBS exchange on the surface of the vesicles, leading to a loss of the functionality, while in the case of the chemisol samples, we have a covalently bound, so we cannot lose the functionalization, and that lead to an increase of the uptake due to the cooperative interaction of the different antibodies bounded to the vesicle surface. And so in conclusion, next slide, please. In conclusion, with this work, we give a first investigation of the role of the protein corona in exogenous surface functionalization of extracellular vesicles, bringing to light an underrated issue in AV engineering and their related application by providing original and quantitative results on the functionalization of VVs with the targeting antibody of relevant cancer therapy as the cetuximab. And in future, next slide, please. And in future, we aim to develop new conceptual and technical methods to handle MV corona studies, including also the study of the composition of the corona, its formation in terms of kinetics and dynamics, its functional effects in biodistribution, and also its exploitation in diagnostic. And furthermore, we also aim to study the mechanism at the base of the AV uptake mediated by the CTX-EGFR interaction. And finally, next slide, please. Finally, I would like to thank all the BioCSI lab from Brescia, led by Professor Paolo Bergese and Professor Annalisa Radeghieri, and all the collaborators of this work, and all of you for your attention and patience for for all the convenience of this presentation. And sorry.
No worries. You know, this happens. It happens regularly. Well, we got through it. So thanks for being persistent there. And
yeah, but when we tried before, it worked.
yeah, right. It doesn't have any problems until we're relying on it, right? So but we got through it, and thanks for that. And we will. So now I am going to allow unmuting. And I just want to remind everybody that if you do have some questions for Annalisa or Angelo about their project here, please put those questions in the chat box, and then I'm going to call on you in the order that we get the comments. So, all right. So now I have allowed unmuting. So when I call on you, please go ahead and unmute. And if you want to share your video, you can do that too. So we're going to start with Maha Jabin. Could you please go ahead and just let us know your question? Thank you.
Hi, Angelo. Thanks for the talk. Really enjoyed it. So my question is about FBS. So can you please repeat your explanation on why FBS help with the EV uptake? I'm asking this question from the point of view that FBS is like a black box and it's not completely defined. So is there any particular mechanism that you think is helping with the uptake? Also, I'm assuming that the FBS you used was the EV-free FBS, not like regular FBS, right?
Yeah. Yeah, sorry, can you repeat your question? I didn't get it.
Can you hear me?
Yeah, I can hear you, but can you repeat your question, please?
Yes, there's some static coming from your end, but I'm going to repeat it anyway. So FBS, it's the serum, right? Fetal bovine serum.
Yeah, yeah.
I was thinking you showed that with FBS, the EV uptake increased in the cells. Is that correct?
Yeah, is that correct? Yeah, because probably due to the presence of the FDS, we see a lower uptake of EVs in general, also of the pristine ones, because there is like more competition of different mechanisms of uptake of other biomolecules that are like not saturated but are more busy, and so for this reason, we, in absence of the FBS, when all of these mechanisms are not occurring, we have a higher uptake of our EVs. I don't know if I answered your question, or Annalisa want to add something.
That actually we have a higher uptake in absence of FBS. If you actually have.
Okay, sorry, then I thought it was the opposite.
Okay, no, because actually the cells are more hungry, but we have a higher uptake of chemisorb EVs when we are in presence of FBS. The uptake is higher when we have chemisorb EVs towards instead in comparison with the physisorb with protein corona, because we believe there is in the presence of FBS, protein composing the FBS exchange with the cetuximab that is just weakly absorbed on the surface of the vesicles. So then basically the cetuximab is not anymore there, and it doesn't drive the interaction of the vesicles with the membrane, cell membrane, while keeping it covalently linked. Then this favors the entrance, but in the presence of FBF, we see always a lower intake.
Yeah, now it makes sense. Yeah, I understand now. Thanks.
Sorry, thank you.
Yeah, thanks for the question. We next go to Christina.
Hello there. Beautiful talk. Thank you. I'm actually in the middle of writing up a paper looking at amyloid on exosomes, so this is perfect and timely. I have a question. So when people do BCA assays versus looking at, say, EV numbers to use as a normalization to treat cells, if we do have this corona, and if BCA assays, for example, are based on, your standard curve is albumin, and if we have a lot of albumin on EVs from plasma, what are BCA assays helping us with, or are they useful at all, or should we be always using EV numbers? Because I default to EV numbers, but I end up having this conversation multiple times with people about, we can't normalize for all the protein on the surface, and the BCA assay is going to be skewed based on that versus numbers. But maybe I'm incorrect, and I would love people's opinions.
Angelo, would you want to?
Yeah. In this case, for this work, we when I show you before the characterization of our vesicles, we assessed particles per protein ratio obtained by NTA and BCA. And then we also checked for this ratio after the functionalization with the antibody in order to check if the BCA is affected by the presence of the antibody. But we also try to subtract the amount of the antibody that we quantified by fluorescence from the one that we obtained by BCA to relate with the vesicle number. And I can say that in our case, the presence of the antibody is not so relevant for the BCA, don't affect so much the measure for the when you go back to relate the BCA number to the vesicle number. But we double checked everything. So we check also by NTA and BCA always to verify if the ratio is constant or not. Yeah.
So in every separation technique that you use, you basically, every time you change the corona that is on the vesicles, because you treating differently the vesicles, then the protein that are absorbed on the surface of the corona will change. So just dosing and characterizing and just numbering the vesicles by simply looking at the proteins may be misleading, I think. So it could be, as also the MISEV guidelines have declared, I think it's better to normalize either protein to lipid ratio every time, or always to detect the AV numbers by some NTA or similar technique. So
Perfect. Thank you.
Welcome.
Thanks for the question, Christina. Next, Massimo. So maybe I'll just read this then. Did you validate the exchange of the antibodies due to the presence of the FBS? And the comment here is that protein corona can hamper.
Yeah, sorry about that. I was in a
Oh, you're
mute.
there. There you are. Go ahead.
Yeah, I'm sorry about that. So I forgot to be on mute. So excellent job as usual. Excellent presentation with some problems due to the distance. But I have basically two questions, two curiosities. I would like to know, your opinion about that. The first one is more technical. So you said that, in your opinion, there is an exchange between the antibodies absorbed and the FBS. That makes a lot of sense to me. Did you validate it, like experimentally, that, for instance, that you have no...
We have preliminary data that show that it's actually exchanged, but just preliminary data. Yeah, we are validating.
You have less after that you incubate with FBS. Basically, you have less antibody on you. Yeah, makes a lot of sense to me. The second question is much more, it's a little bit puzzled me what you said, that basically if you introduce your EVs with physisorb antibodies, the FBS is able to exchange, therefore to affect the ability of your EVs to interact with the cells. Whereas when you have chemisorbed, so basically covalently bound antibodies, basically the FBS is not able to affect. But if you remember by Dawson and Monopoly, they published that very famous Nature Nanotech paper saying that the corona is able to cover and basically to hamper the ability of targeting agents to do what they're supposed to do. And then, 10 years ago, there were so many papers with people solving this problem by, PEG chains, trying to make the targeting agents to protruding out from the surface of the synthetic nanoparticle. So in your opinion, why didn't you see the same phenomenon on the extracellular vessel? It's really interesting, but why is that, in your opinion? You understand the question, Angelo? So it's... I was surprised, if you saw it, it means that something's happening that makes probably the protein corona of an extracellular vessel somehow different than the protein corona of a synthetic nanoparticle. You know what I mean? But why?
So if I may speculate, you can tell me if I'm off base here, but as I understand it, this might be the difference between the soft and the hard corona, where it's the hard corona where you actually have proteins that are kind of denaturing, coating the surface of the synthetic nanoparticle, that may be a bit different for EVs, where there's more of this loose association. What do you think about that, Angelo and Annalisa?
Yeah, I think that it's... I have to think about it. And
I can start if you want.
Huh?
I can start if you want to say my opinion.
Yeah, you can start.
No, I can raise a very interesting point, because we really need to start to know a little bit more about the vesicle and their surface, and because since all the research has been so far on synthetic nanoparticles, so rigid, hard, and with all these very similar properties, while vesicles are all completely different nanoparticles. So that's an interesting point. The other fact that we see that FBS doesn't affect the interaction of covalently bound cetuximab to the membrane. Maybe it can be due also to the PEG. So we actually the cytosine is linked to PEG units.
So there is a PEG chain.
It's a PEG chain. So
Yeah.
Okay.
Indeed, we don't see any corona basically physically absorbed on the surface of covalently bound, which was kind of astonishing to us. So when you do the washes, then it simply doesn't change the concentration of the number of the antibodies that are linked to the surface of EVs. And we think this is our hypothesis. We are investigating. Angelo is in Spain for that. So we are investigating this fact that maybe adding a PEG moiety, then it changes the way FBS proteins absorb on the surface of the vesicle, like it has been demonstrated for synthetic nanoparticles. So maybe it could be that this is.
So how long is the PEG chain? Because this is actually.
Yeah, the PEG chain that we use is long, is 4 units. It's not very long.
Yeah, interesting. I'm extremely surprised.
In my experience, for now, I see that actually the PEG changed the surface. If you bound a small PEG to the vesicle surface, you are changing their surface properties. And also, if you use also different vesicles and we put the same PEG chain in different vesicles, you are like almost normalizing the surface with the pegylation. So yeah, actually the DV that you are producing in this way are no more natural, are more synthetic, and you have modified the surface. So the protein corona, it's due to the contribution of the environment, but also of the surface, because it's due to the nanoparticles environment interface. So also the surface of the particles is important. And yeah, for this reason, also the pegylation, I expect that also the pegylation changed the surface properties of the vesicle.
Can I ask you the last question? So basically, you pegylate the vesicles, but you think that you didn't use all the PEG chains on the surface. So for this reason, it would make sense that basically you decrease somehow the absorption of the protein corona.
Yeah, I don't expect to use. Yeah, we didn't demonstrate it yet that we don't use all the PEG chain that we bound to the vesicles, but I don't expect that we use them all, because also for steric hindrance reasons, for the antibodies, big macromolecules, and yeah, probably we have to check for this, how many PEG are effectively bounded with the antibody on, and how many are still free to exert antifouling properties on the vesicle surface.
Thanks, Angelo. By the way, exciting what you have now. Thanks.
Let's see. Are you both able to stay for a couple more minutes? I think we have 2 more questions here, and then we'll cut it off. Okay. Sheila, please go ahead.
Hi, good for you, Angelo, for plowing through your talk through like technical difficulties. That was really challenging, and you did very well. My question is, has anybody tried to visualize the corona? Because there's like really good EM techniques now that can look at like individual like phospholipid heads at the moment, and it's an exciting time for EM, and I'm wondering, has anybody in the field applied this to look at the corona?
Angelo?
Yeah, well, so I guess there has been some EM and also some AFM that's been done.
Yeah, also sure.
And also EM, also with EM microscopy, they have seen the lipoproteins interacting. So some groups, Sara Busato's groups, and Joe Wolfram, and also another group that I don't remember now, but there was another publication cited.
Yeah, I shared that too. Yeah.
So they have seen interactions. So, but with bigger particles, because it's hard to see the corona of like single proteins, basically. But there are some works that are doing this.
Yeah, I think the EM technology has advanced considerably, and I think you can see individual proteins. Like, so I think that might be something to investigate.
Thank you.
Yeah, thanks for the question. And final question, Andrea.
Hi, thank you for your great presentation. I was just wondering on the PEG question. So PEG is also used for LNP synthesis, right, in lipidated or pegylated lipids. And it's actually known that the shedding of the PEG molecules from the lipid nanoparticles is an actual issue for the delivery of, or efficient delivery of the particles once it is in vivo. I was just wondering why this shedding happens so fast in LNPs and it doesn't seem to be happening on your functionalized EVs.
We don't know. This is yeah, and the word of synthetic nanoparticles, I hope it will help us, even if we are looking at very different types of nanoparticles. So it's extremely challenging to look at this. So thanks really for the question. We at the moment, we don't know. We don't have an hypothesis for that. So I don't know if you can help us maybe.
Yeah, I think that's a great stopping off point for our session today. That there's a lot of knowledge that's been accumulated in the synthetic nanoparticle world. We're just starting. Just this EV corona field is just a few years old now. So thank you, Annalisa and Angelo, for your massive contributions here and for sharing with us today. And we'll certainly look forward to how things are developing over the next few years, and hopefully with some good collaborations with folks from the synthetic nanoparticle world too. So thanks again, and thanks everybody for joining. I'm going to put the results of our little poll on X and LinkedIn so people can just see that for fun. Thanks everyone. Take care.
Bye. Thank you.